Hello, everyone. Thank you for joining us for CEN's second annual virtual symposium, Advances in Drug Discovery and Development. I'm Mitch Garcia, an assistant editor at CEN, and I'm delighted to introduce our next speaker, who is Hagop Youssoufian from BIND Therapeutics. Dr. Youssoufian has served as BIND Therapeutics Chief Medical Officer since November 2014, and additionally has led regulatory affairs and quality assurance activities since March 2015. Dr. Youssoufian will be presenting a talk entitled Developing Accurate Nanomedicines, the BIND Therapeutic Approach. I'll be back to moderate a Q&A session immediately following the presentation. We welcome you to ask your questions at any time. Just click the green Q&A button in the left corner of your screen. And if you need any technical assistance during the presentation, please click on the support button located top right of your screen. Please note that CEN does not endorse any company, products, or services that may be mentioned in this presentation. Finally, this talk will be archived and available on demand at the conference website for 3 months after the live broadcast until December 16, 2015. And now I'm delighted to hand it over to Dr. Youssoufian.
Well, thank you very much, Mitch. I appreciate being able to present you some data from our company, BIND Therapeutics, and equally share some ideas around nanomedicines and nanotherapeutic development, which I certainly feel is a significant opportunity to bring something unique to patients suffering from a variety of illnesses. In our own case, our focus is clinical oncology, but in fact, nanomedicines have numerous potential applications, oncology being just one. But the examples that I'm going to use today are based on our own experience, primarily in the oncology realm. So BIND has been now a public entity for several years. We are a clinical stage company. Our sole focus is the development of specific, targeted, I dare say unique and proprietary nanoparticles. We call them Accurins. And so a schematic of that is shown on the first slide. But let me go on to first remind you that I will be making forward-looking statements in this presentation, which for a full description of it, you are welcome to consult our SEC filings. And with that obligation out of the way, let me start with our presentation. So nanomedicine development pathway has a number of similarities to the development of small molecules, the development of biologics. Conceptually, it's the same pathway of identifying something that one thinks would make a difference in terms of the biology of the disease, and also identifying an important unmet medical need in which to apply the discovery and that knowledge. Where nanomedicine perhaps differs from some of the other disciplines is that certainly it's a convergence of lots of different sciences, chemistry and engineering being two of the more prominent ones. But with regard to relative importance, we, at least at BIND, emphasize engineering and the way in which we make our nanoparticles, the size, some of the surface charge characteristics, the diffusion properties, are all fundamental engineering issues that are more relevant to nanotechnology development than chemistry per se. Chemistry is still quite important, but at least in our case, we don't necessarily attempt to optimize the API or the drug product that is embedded in the nanoparticle. That we assume has been done to some extent already by our partners, and so what we bring to the table is the ability to first of all, improve some very basic pharmacokinetic characteristics of a molecule that may suffer from those kinds of challenges, and make its biodistribution much more predictable, improve the therapeutic index by limiting exposure to healthy tissue, and specifically, or more specifically, concentrating a drug at the site of disease. But also, quite importantly, nanotherapeutic development in nanomedicine is not a very new idea. It's been around for a number of years, and it's been pursued by many very prominent academic groups, and also in certain companies, where we think we can make a difference on the industry side is with process development and scale-up. In the academic world, many innovative directions are being taken, but to operationalize it, to scale it up so that one can conduct clinical trials, eventually lead the drug toward approval in one or more indications and commercialize it, that requires the very disciplined process development approach that companies such as ours bring to the table. So again, to emphasize that this is a multidisciplinary effort, but at least in our case, engineering plays a particularly prominent role. So one of the most challenging issues in any drug development endeavor is to, again, to control the biodistribution of the therapeutic agent, to amplify the presence of the drug in site of disease, and to minimize the exposure of healthy tissue to a potentially toxic agent. So here's a contrast of sorts. With a conventional oncology therapeutic, if one were to administer this, and it doesn't really matter how, but let's say intravenously, a drug like the payload that I'm going to be discussing very shortly, docetaxel, distributes widely, so it has a high volume of distribution. And the circulating half-life is also very short. And it's not necessarily the case that docetaxel is enriched in the tumor. It also goes to the kidney, liver, lung, etc. So invariably, there's a significant amount of exposure to healthy tissue. And this is primarily the problem that a very effective drug like docetaxel presents to oncologists, in that although it is effective, the therapeutic window is relatively narrow. And so what we hope to accomplish with our nanoparticles, and we call them Accurin, is to effect a more targeted delivery and enrichment around the site of the tumor. Now, you can see on the right side of this figure that there is still some green dots that are distributed systemically, but the difference is possibly an order of more magnitude difference. So there's a great enrichment within the tumor and a relative sparing of healthy tissue. What that also does is to increase the circulating half-life of the drug, so that there's more of a continuous bathing of the tumor with a nanoparticle-loaded drug. So what are Accurins? Accurins are polymeric nanoparticles that have several very important components. And one comment, again, to underscore is that virtually everything we use at BIND Therapeutics has been in patients already and has been validated to some extent, so that the risk of development is somewhat lessened by virtue of the fact that those are validated components. And what we aim to do is to use polylactic acid, or PLA, which is an FDA-approved polymer that has been in patients, and after infusion, the polylactic acid degrades to lactic acid, and it's cleared by the Krebs cycle. So normal metabolic processes take care of that particular component, and it's nothing unusual that the body hasn't seen before. So this is an FDA-approved polymer, and what it does for us is that it allows us to entrap a payload. I mentioned docetaxel several times, but it doesn't have to be docetaxel. It could be a variety of other biologically interesting payloads. The entrapment in this case is not covalent, so we are not modifying docetaxel or any of the payload components per se, not modifying them chemically, not to coupling it to the PLA, except in this non-covalent way. We also take pains to engineer the nanoparticles so that they are leaky, but not in an unpredictable way. So there's a regulated release of the payload over days. The second component is the therapeutic payload, or the API. And again, this could be a broad range of payloads. The first entry to the clinic is BIND 014, which is loaded with docetaxel, but it could also be peptides, proteins, nucleic acids. So there may be a number of different payloads and a number of clever ways of delivering therapeutics that have the challenges that I alluded to previously. There's, again, it's important to note that there's no chemical modification of any of these payloads, be it a small molecule or a biologic. The third component is polyethylene glycol. Now, there's actually a very important reason why we use PEG to coat this PLA that is now containing a drug, and which is the following: that if we left it on its own without coating it with PEG, that looks like something that the immune system could clear it very readily. It's a hydrophobic entity, and it's easily recognized by immune cells and cleared very quickly. The PEG introduces a hydration shell around the particle, and it makes it look like a water molecule, essentially, so that it stays in the circulation much longer, and it's also now protected against immune clearance. And the fourth component that is unique to the platform that we work with is to introduce a targeting ligand for targeting to cells that express a particular receptor, and this can lead to enhanced particle accumulation at the site of the disease. It could lead to cellular uptake and internalization of the nanoparticle, although, and this is a very important distinction from antibody-drug conjugates, this is not a requirement at all. The targeting ligand receptor system could be, we have many choices, but the one that is in clinic now is a PSMA ligand, which is also a ligand receptor pair that has clinical validation. So the key points that I'd like to underscore here is that the nanoparticle is made up of several component parts, all of which have been validated, and together they constitute essentially a new molecular entity that we hope will be more than some of its parts. So challenges with small molecule drugs that can be addressed by nanotechnology are the following. In the small molecule world, having something that is a very high molecular weight or is hydrophobic or insoluble, usually that sort of a molecule is destined to not being advanced. It's probably going to end up in the dustbin. So it's not an ideal profile for a conventional drug candidate. But this actually makes it very attractive for us, and it's a suitable payload for an Accurin. Why? The relatively large size of the molecule ensures a continued presence within a nanoparticle as opposed to a very rapid leakage from the nanoparticle. The hydrophobic nature of an API makes it compatible with the lipid, the lactic acid moiety of PLA. And lastly, if a drug is insoluble in circulation, the nanoparticle entrapment overcomes that, and once the drug reaches the right environment, then access to tumor tissue or to diseased tissue is enhanced so that the solubility during circulation is no longer relevant. So those are three major challenges in traditional drug development that can be addressed by nanotechnology. But that is not to say that the bad old drugs are the only ones that we can actually work with. So our scientists have also figured out ways of really broadening this therapeutic window that I mentioned earlier with payloads that have more favorable intrinsic characteristics. So, for example, if a drug is not, terribly insoluble, or if it's not very hydrophobic, but it's cationic, well, to make it enter and be compatible with PLA in that hydrophobic environment, what our scientists can do is to introduce a counterion, and so now it becomes a neutral hydrophobic ion pair. So this is also something that we have published, and with some of our collaborations with pharma partners, we've been able to generate these kinds of nanoparticles. So again, there's no requirement, no need for chemical modification, and it leads to the development of a molecule that is good, but perhaps not good enough, and it allows us to take advantage of the targeting characteristics of a nanoparticle to further improve that therapeutic window. So there are two fundamental mechanisms through which we target nanoparticles. One is a mechanism called EPR, or enhanced permeation and retention. And this is something that applies to liposomes. It also applies to nanoparticles. and it has to do with the large pore sizes and the leaky vasculature that are part and parcel of the tumor microenvironment. So nanoparticles gain entry into the tumor environment by more readily escaping through these leaky vessels. So that's EPR, and the way we think about it is that this is fundamentally tissue targeting. Now we go a step beyond that, and that actually is achieved by taking advantage of well understood receptor ligand pairs to engineer our nanoparticles. And as I mentioned, our first entry in the clinic was with the PSMA targeted ligand. And in fact, here in this xenograft model, we show the same tumor on one side, which is designated by that negative symbol. This tumor, which is a prostate cancer tumor, is not expressing PSMA. So the particle still shows differential accumulation in the tumor that is not expressing the target of interest. However, if we knock in that target, which is PSMA, and that's shown on the contralateral side with the plus symbol, now we see even a greater accumulation of the nanoparticle. So that allows us to say that both the EPR effect and specific ligand receptor targeting are both relevant and desirable with certain payloads. This may or may not be necessary with all payloads, but again, our first entry in the clinic leverages both tissue and ligand and cellular targeting. So the engineering approach that we follow here uses a variety of dials to establish the optimal particle size, the ligand density, and the API load. But of course, it doesn't stop there. So our engineers can generate several different versions of a nanoparticle. Then it's truly a collaboration with our biology group to figure out which one and which particle with certain diffusion characteristics gives the best outcome in our animal models. So far, that is somewhat predictable, but also empirical. So we actually end up testing sometimes 3 or 4 different nanoparticles before going back to the engineering group and saying that make more of this, this is what we want, this is what we want to nominate for development. On the toxicology side, one might think that there would be differences in the toxicity too of different nanoparticles with different diffusion characteristics. What I can say clearly is that the empty nanoparticle by itself is actually quite benign. There's very little toxicity associated with it, if any. So the empty nanoparticle is non-toxic. So whatever toxicity we observe is therefore directly attributable to the payload and the diffusion characteristics that are embedded in that nanoparticle. Well, so the way we think about nanoparticle development here is that with our first entry with an approved drug, we have an opportunity to differentiate it from the parent drug. And we also are in a very good position of working with a validated drug. We know where to go in clinic, which tumor types to pursue. So in a sense, this is a differentiated efficacy with approved drugs. The next stage that we're exploring now is to allow us to probe other pathways with inhibitors that may be highly potent. They're generally ones that involve well understood mechanisms in cancer biology, but their toxicity may be severely limiting and therefore resulting in a very narrow therapeutic window. So this is what we'd like to overcome. And thirdly, we're also now exploring breakthrough therapies, so to speak, therapies with oligonucleotides as payloads, therapies in the immuno-oncology arena, where we can also utilize the kinds of targeting ideas that we've been learning through our current efforts. So to talk more about where we are now in clinic, let me just take you through BIND 014. This is, again, the PSMA-directed nanoparticle that is loaded with docetaxel. So you can see two examples here of our attempts in that first bucket with known and approved drugs. Docetaxel is the one in clinic. We've also made inroads with vincristine, which is a microtubule inhibitor, somewhat similar to docetaxel mechanistically, but not exactly. And both are very potent drugs, but the toxicity can be prohibitive. Here, you can see that with BIND 014, our clinical candidate in a model versus docetaxel, there is very good activity with the nanoparticle, better than with docetaxel. And with BIND 510 is the nanoparticle loaded with vincristine. There's also improved activity with the nanoparticle version versus the free drug. In the panel below, you can also see PK data of BIND 014, and the prolonged PK lasting over days is also a characteristic of our nanoparticles, and sometimes the PK differs. So in this case, 014's PK is different from 510's PK, but that is also something that now needs to be juxtaposed to the biology experiments to determine which one yields the most optimal biological effect. So to take you through the clinical development, to date we have probably treated over 160 patients with BIND 014. Initially, this drug went through a dose escalation study, and the dose that we identified for phase 2 development was 60 milligrams per meter squared, administered every 3 weeks intravenously. It was actually important for us to identify that particular schedule of every 3-week administration, because that's how docetaxel is administered in clinic. And we thought that in the future, if we need to do a head-to-head comparison, it would be important to minimize the variables. So at the same time, we had studied a weekly schedule, but we decided to advance the every 3-week schedule for that reason. But also importantly, with the every 2-week schedule, we began to see some responses, and sometimes very interesting responses. So for example, we had a patient with cervical cancer who, after a number of prior therapies, had a complete response, complete eradication of all the tumor deposits that she had. So this was a very gratifying outcome for us. But we also had responses in patients with lung cancer, prostate cancer, and biliary tract cancer. Now, many of those tumors, in fact, do respond to docetaxel. So from that perspective, it was not a huge surprise that we should see we should see activity in those tumor settings. But nonetheless, this was the first opportunity for this novel targeted nanoparticle to be tested in patients. So we were already seeing activity at this very early stage. We also saw a number of stable disease in a number of patients, which is also an indication of clinical activity. Now, I won't dwell on the pharmacokinetics, but suffice it to say that the AUC is much improved versus that of free docetaxel, and it's also very dose proportional. So all of the behaviors inside the body as the nanoparticle is circulating is very predictable. And generally speaking, at any point in time over the course of, let's say, days to a week or more, about 90%, 92% of the circulating drug, in this case docetaxel, is actually encapsulated. So if there's any free circulating docetaxel, it's in the minority. Now, one of the proof of concept opportunities for us was to understand the targeting aspects that I mentioned before, and the dual targeting mechanism via EPR effect and via cellular targeting. So we're still doing a lot of work in this area, but we are, I think, increasingly appreciating that the tumor vasculature is important for targeting nanoparticles. It's important and perhaps quite intuitive in terms of the EPR effect. But in terms of PSMA expression, there's a fascinating story here. So PSMA was initially discovered in the context of prostate cancer, and that's where actually the name comes from. So prostate cancer cells per se express PSMA. But where we're seeing activity is outside of prostate cancer, and it turns out that rarely is PSMA expressed on tumors like lung cancer tumors or cholangiocarcinoma that I mentioned previously. However, the neovasculature associated with those tumors and the endothelial cells lining up those tumors are actually PSMA positive. And so that is, in fact, we think what's happening here, that the nanoparticle is going to the right place in the tumor and being targeted to the tumor through both the EPR effect and through the presence of PSMA on tumor-associated vasculature. So based on that successful outcome from phase 1, we then entered phase 2 development, and we tested the drug in an open label study in 40 patients with second-line non-small cell lung cancer. The dose and schedule of the drug that we used came exactly out of our phase 1 study. And in this study, we were also very interested in first understanding whether we have a drug here for further development, but also if we could further validate the platform from a clinical standpoint. And we think that we are well underway here, so let me take you through some of the phase 1 data. We saw a clinical activity in this tough-to-treat patient population. So specifically, we saw 6 partial responses, or 15% overall response rate among 40 patients. Now, we believe that that is meaningful because the typical response rate with docetaxel is in the single digits, let's say 5 to 8%. And we were seeing these responses despite reduction of 20% in the dose of the payload. So docetaxel typically is administered at 75 milligrams per meter squared, whereas the dose that we're using with our nanoparticles is 60 milligrams per meter squared. So despite that 20% reduction in the dose, we're seeing activity that we think is above and beyond what we should be able to see with docetaxel. Very importantly also, the tolerability of nanoparticle appears to be much better than that of the parent drug, or free Taxotere. So if I could call your attention to the table on the right side of the slide, we're comparing adverse events in this trial with BIND to the adverse events in second-line lung cancer patients who were treated with Taxotere, and those numbers actually come out of the Taxotere FDA-approved label. If you look at hematologic toxicities, for example, anemia, any grade, 20% with BIND 91% with Taxotere. So a substantial difference, no statistician needed, I suppose. It's also the case for other hematologic toxicities and for some non-hematologic toxicities as well. Now, the criticism that could be made with these data is that these trials were done, several years apart. There could be differences in supportive care, all of which is quite legitimate. But if you scroll down in that same table to alopecia, or hair loss, with BIND 15%, with Taxotere, 56%. And of course, there's no supportive care involved there, so it's a clean readout of what's going on at the healthy tissue level. And this is the kind of data that gives us a lot of excitement and hope that the platform is performing as we had predicted. Namely, it's resulting in the substantial activity that I mentioned before, but it's also sparing healthy tissue. Now, since probably only some of you may be experts in lung cancer, let me just take a sort of quick interlude to say that the lung cancer space, treatment, the classification has become quite sophisticated now. It's based on histology, it's based on molecular characteristics, so it's no longer a single disease entity. So even though our trial was done in a population that is essentially a real-world patient population, we were actually very pleased to see that in 2 large subgroups of lung cancer, those with KRAS mutated tumors and those with squamous cell histology, there was also substantial activity. And both of those subtypes are ones that are notoriously difficult to treat. In fact, for KRAS mutant tumors, there's no approved therapy specifically for that genotype. For tumors with squamous histology in the second-line setting, until a few months ago, there was nothing specifically approved. Now, with Opdivo, which is BMS's anti-PD-1 antibody, there is fortunately something available to patients, but it's only the minority of the patients that benefit from that therapy. So there's still a lot of room for improvement. And here's the data that I just alluded to. Let me just make one other point here, which is that our trials so far have been single-arm trials, so these are not randomized trials. Therefore, the comparison is invariably to historical data. But there's also very good historical data. So with KRAS mutant tumors, another drug that is now in phase 3 development compared the combination of that drug to docetaxel versus docetaxel. And the response rate with docetaxel was 0%. What we saw in our case was a total of 3 responses in 9 patients. So we're quite intrigued by that. But we'd also like to continue our study in KRAS mutant tumors so that we'll have more confidence in taking next steps and developing a pivotal trial in that setting. In the squamous histology subtype also, we were very intrigued by the overall survival outcome of 11.1 months, which is quite interesting, especially compared to the Opdivo trial that I mentioned that just received approval. In that case, Opdivo was compared to docetaxel, and the median survival on docetaxel was just around 6 months, which is not that unusual. The additional importance of these data is not so much that we are getting clinically superior results, but perhaps even more importantly, that the platform really is performing as we had set out to as we had anticipated, namely resulting in greater activity versus docetaxel and lower toxicity. Now I'm going to take you quickly through some of the next steps that we're doing. So it's not only cytotoxics that we can engineer in nanoparticles, but it's also a variety of very interesting kinase inhibitors, some quite potent, such as the Merck compounds. We have a very interesting collaboration with them. There are 2 agents in this collaboration, a KSP inhibitor and PLK1 inhibitor. Both KSP and PLK1 are involved in cell cycle control. And through a different collaboration with AstraZeneca, we engineered nanoparticles containing an Aurora B kinase inhibitor, which is now being readied for phase 1 development. So those are very exciting opportunities for us to learn more from our platform and also to provide something valuable to our industry partners. And here's one example with the Aurora B kinase inhibitor. On the left, you see that the parent drug versus the control virtually wipes out the bone marrow. So the blue cells are gone, and that is a classic toxicity that results from Aurora B kinase inhibitors. But with the Accurin, it's actually almost indistinguishable from what's going on with the control panel, especially Accurin 2. So again, to the earlier point that it's protecting healthy tissue, we believe that this is also the case here. And on the right side, one of several efficacy experiments where Accurin 2 shows in fact, some tumor regressions versus the parent drug. So again, very exciting preclinical data to position it for clinical development. Also importantly, more recent studies that were presented at AACR showed activity in several other models, including a lymphoma model and a small cell lung cancer model. But on the top panel is where I'd like to call your attention. You can see that with the parent drug, with the naked aura kinase inhibitor, the drug doesn't really accumulate very effectively in the tumor, and it's essentially not there by 24 hours. Whereas with the Accurin formulation, we see drug essentially almost uniformly throughout the tumor 9 days after completion of the dose. So this again speaks very powerfully in our minds to the targeting and the retention of the drug within the tumor environment. So just to make that point a little bit more, in terms of the pharmacokinetics, we work with several Accurins, and we take the one that, we take several actually to in vivo models. So on the right, you see a breast cancer model where with Accurin D, there's a lot more API in the tumor. On the lower left-hand side, this is a very interesting pharmacodynamic marker, which actually can be dose limiting for this class of drugs, where you see the red line showing a vast increase in glucose levels after treatment, but with the Accurin, that is essentially completely ablated. So again, this may very well be a Cmax effect that is completely addressed by the nanoparticle approach. And the last concept that I'd like to talk to you about is breakthrough therapies. Now, of course, there are a number of very interesting additional applications with Accurins, and what we're doing is shown on this slide. We have a collaboration with Macrophage Therapeutics, and it's in the immuno-oncology space. Now, of course, there have been substantial inroads into our understanding of checkpoint inhibitors, PD-1, PD-L1, and so on, but not nearly as much of an understanding in terms of the tumor microenvironment and the various cell types that play very important roles in immunosuppression. So one of the key cell types in the tumor microenvironment that's involved in suppressing host immunity is the macrophage. And the story can get here quite complicated, so let me try to give a relatively straightforward version of it. There are several different macrophage types that can be polarized and can go from one direction to another. Some of those subtypes are pro-tumoral, so they're obviously bad. Others can be anti-tumoral. And the idea is that if we can change the pro-tumoral macrophages to become anti-tumoral, that would address a very important component of the tumor microenvironment and allow us to bring something unique within the immuno-oncology area. So what we're doing is that we established a collaboration with Macrophage Therapeutics. Macrophage Therapeutics is a very exciting partner of NVIDIA, and NVIDIA had an imaging agent called tilmanocept, which is FDA approved, and the imaging agent is one that targets CD206. Now, CD206 is also known as the mannose receptor, and it's present on activated or bad macrophages. So we have established this collaboration with the idea of engineering our nanoparticles with mannose receptor-directed ligands. And we're at the earlier stages of this collaboration, but this is a very exciting opportunity because for the first time, it will allow us to directly target those cells with payloads, and you can probably think of several potential payloads, but what we're interested in is to test them empirically, so either kill the bad macrophages through direct targeting, or directly target the bad macrophages, but allow them to be forced toward the direction of becoming anti-tumoral. And so those experiments are underway, and we think that our nanotechnology and the Accurin platform can bring something special to the table. So in conclusion, this is a big effort, and of course it involves collaborations internally and with several of our current partners, and we're very fortunate to be collaborating with them. The versatility and the scalability of our Accurin platform, we think, are very important differentiating points. It allows us to introduce multiple different therapeutics, including small molecules, nucleic acids, and proteins. It allows us to target specific cells with ligands that may be small molecules, but also with larger molecules, peptides, and antibodies. And although I've been discussing cancer biology throughout my talk, this approach is also quite relevant to other inflammatory conditions, vascular diseases, infectious diseases. BIND 014 is our first product to enter clinic. It's a novel PSMA targeted nanoparticle that contains docetaxel. Certainly we believe that the greater efficacy and tolerability that we've seen with BIND 014 versus docetaxel allows us to say that the therapeutic window is being affected in a meaningful way, but also that the platform is working. So our initial and we believe promising data has now led to additional trials. These are multi-center phase 2 trials in both lung cancer and in other tumor types where we identified that additional tumors that have limited that can be impacted with docetaxel, but in a limited way. We now have the opportunity to bring something even more potent and to impact those tumors. And lastly, our collaborations with AstraZeneca, with Pfizer, with Roche and Merck, and with Macrophage Therapeutics allows us to broaden our bandwidth considerably and go beyond the data that I showed to you already into some other very exciting areas. So thank you very much.
Okay, thank you, Youssoufian, for the talk. There's several questions have come in, so I will ask the first one. I'm going to synthesize two questions that are very related. This question was from Tarek Fadel from NNCO and from William Litchfield. What is the typical size of these nanoparticles, and how do you ensure that the loading of the payload is done homogeneously in such a way that each nanoparticle has more or less the same amount of API?
Okay. So the typical size of our nanoparticles is around 100 nanometer diameter. And the way we ensure that there's optimal loading of the nanoparticle with the payload is really that I forgot which number slide it was, but our engineers are very, very good at gauging different payload densities, and so some of this then is handed over to the biology group. So it's really a collaboration as to what is that optimum profile at the end. But I would probably say that with docetaxel, there are, on the order of several thousand molecules per nanoparticle.
Okay, so another question. This is from Chris Brown from Evotech. Can the nanoparticles target the CNS or pass through the blood-brain barrier?
Well, it's a fascinating question. So it's a terrific question because the nanoparticles are large molecules. I don't think that they can readily go through the blood-brain barrier, except that the neovasculature that is also quite relevant to a tumor like glioblastoma can be accessed, potentially can be accessed with a PSMA-directed nanoparticle. But in addition, one can also think of other clever approaches with brain shuttling molecules, for example. And so there's a lot more to be done in that area. And stay tuned. It's work in progress.
So another question about your nanoparticle and technology system. This is from Shen Shi from Chatham University. Could the technology be developed to work with more metastatic tumor types, such as lymphoma or melanoma?
So I personally think that there's very little out there that is beyond the limits of applying, really clever biology and engineering approaches. So there are certain lymphomas that I think could be accessed. And one piece of data that I showed before was in a model called diffuse large B-cell lymphoma, or DLBCL, through our collaboration with AstraZeneca. So that has not yet been tested in patients, but that is the kind of proof of concept data from preclinical models that gives me hope that the same would happen in a clinic. Melanoma, likewise, I think should be amenable to those same approaches.
So the next question, I'm going to do my best to synthesize this as one, as two people had the same question. This is both from Erwin Russell from Princeton Institute of Technology and Patrick Schell from Merck. The question is, how stable are these nanoparticles with respect to such as particle size and APA leakage, and in what environment are they kept in order to keep their stability intact?
So again, part of the optimization steps that I discussed previously involve all of these aspects: stability, how fast the payload diffuses out of the nanoparticle, the pore size, etc. So I'm afraid there's not a single answer, but in general, those nanoparticles are very stable. On the clinical side, I can tell you that we are doing multi-center global trials, and at least on the clinical side, these nanoparticles are really quite user-friendly.
So this will be the last question I have for you today. There's been a lot of questions about the advantages of using a targeting ligand versus a non-targeting ligand. I know you had that one slide showing the difference comparison, but I think people would like if you could elaborate a little bit more on that. And also, what do you feel is the advantage of Accurin in comparison to Abraxane? Is targeting ligand PMSA truly important for what's been observed?
So it's a great question, and again, it's not a one-size-fits-all proposition. In the case of BIND 014, we believe that PSMA targeting is actually playing an important role in tumors outside of prostate cancer. Now, with regard to Abraxane, actually, Abraxane is not a targeted particle, but the PEG coating, quickly comes off after infusion. It's basically there to obviate the need for chromophore, which is a solvent, let's say, that is associated with hypersensitivity. So that's all it's doing. In our case, the PEG is forming a protective shield and improving the biodistribution of the nanoparticle. So conceptually, it's different. Also, although with liposomes, one can generate immunoliposomes, but it's generally very difficult to introduce targeting components on liposomes. With our technology, although there are several components, it's actually all assembled in a single step. So this is part of the intellectual property that we have that makes it very, very difficult from some of the other approaches. So I hope that at least partially answers that very good question.
So that's all the time we have. Many thanks to Dr. Youssoufian for your presentation, and thanks to you all for tuning in. There'll be more great talks beginning at the top of the hour throughout the rest of the day. You can find the full program on the agenda page located at the upper right-hand side of your screen. In the meantime, please stop by the virtual exhibit hall and the lounge to check out our sponsors and exhibitors or network with other attendees. Join the conversation on Twitter with #CENSIM15. And if Dr. Youssoufian has anything last parting comments to say, I'll pass it over to him.
I'd just like to thank you all for the opportunity to present to you these data. We are certainly excited by our platform, but also very excited that we can provide, I hope in the near future, something meaningful to our patients. So thank you very much.
All right, thank you, and that will conclude the talk for today.